Hey again guys, so yeah, I was right, it is taking forever to upload, so I'm just going to do lots of short videos and hopefully they'll upload a bit quicker for you. Sorry about that. Okay, so question 4. A group of students was given a 1% solution. That just means that's how concentrated it is, not very concentrated. 99% of it will be water. So 1% solution, weak solution of an unknown digestive enzyme. They were also given 3 tubes containing an identical mixture of foods. The students carried out a different biochemical test on each tube before and after adding the unknown enzyme. The results are shown in the table below. So they've all got the same food and the same enzyme. So colour before, so we've got bi-ret, so that's protein. That's a protein test. Starts off blue and then it goes purple. So before they added the enzyme, protein was present. So yes, protein. And after they added the enzyme, it was purple. So yes, there's protein. So it can't be protease because the proteins have not been broken down. So not protease. Next, iodine, that tests for starch. Starts off as an orangey amber colour, and then if starch is present, it will turn blue-black. So before they put the enzyme in, it was blue-black. So there is starch in the little food mixture. After they put the enzyme in, it's yellowy orange. So there's no starch left anymore. It's not disappeared, it's been broken down. So what is the enzyme that breaks down starch? Amylase or carbohydrates? So in the food, in the test tube beforehand, there was starch, and after they added the enzyme, there is no starch. So starch must have been broken down, which means the enzyme must be carbohydrates, amylase. Let's just double check. The Benedict's test, that's for reducing sugars. And that will go a brick red colour if there are reducing sugars present, or an orangey or a yellow or a greeny if there's a small amount of reducing sugars. So there were reducing sugars beforehand and there are still reducing sugars afterwards. So there's no I can't think. Well, there's no enzyme to break down reducing sugars. So it must be carbohydrates because that's the only option that we have. I'm not sure why lipase is on there twice. It's just an error, I think. There might be fats in there. There could be lipase in there, but they say it's only one digestive enzyme. So it's not, we don't know if it's lipase because there's no lipids that have been tested. It's definitely not protease because proteins were present before and after. It is going to be a carbohydrate because it broke down starch. So that was a long-winded way. But I think that was a fairly simple question, so long as you know your food tests. Okay, next one. The figure shows the general structure of an amino acid. So we've got NH2 up here. Now I always think about ammonia, which you might know, I'm not sure. Ammonia and nitrates and all of those compounds, they are NH3, NH4, they have that structure. And NH2 is referred to as an amine group. It's an amine, or we can say it's an amino group or an amine group, however you want to say it. We've drawn them, I think, like at that angle. NCC, remember I got that M&M song wrong. NCC. I actually draw those the other way around, but it doesn't matter. And then we have hydrogens, and then we've got our R group here, which can change, can be different things, which we mentioned before in the first question. And we said that proteins, amino acids, in their R group, it's usually some sort of methyl group like CH3, CH, or a hydroxyl group, OH, and it could contain sulfur. So this is going to be CHOS, a combination of any of those things. It could just be C, could just be H, and so on. We refer to this side, the NH2 side, as our amine or amino group. And then on this side is our COOH, C-O-O-H. Which we refer to as our carboxyl, or carboxylic acid, or just carboxyl group. There's a few ways you can say it. I like saying amino group, carboxyl group, because you've got carb, oxygen, and hydrogen. So the question asks, state the names of the groups labelled U and V. So U, that's our amino group or amine group, and V is our carboxyl group. Again, like I said in the last video, if you didn't know that, use this to make a poster or flashcards or a mini poster or whatever, if that's something that you didn't know, because that is a fundamental. You need to know the structure of an amino acid and what these are called. Okay, so U is our amino group, or amine, either's fine, and V is our carboxyl group, or you could say carboxylic acid. Okay, I'm going to stop it there so I can do the next questions and upload that one.